The overall goal of this procedure is to visualize and quantify the distinct subpopulations of specialized cells that differentiate in biofilms of the model organism Bacillus subtilis. This is accomplished by first inserting transcriptional reporters into the chromosome of B. subtilis. These reporters will be expressed exclusively in the subpopulation of cells under study. The next step is to promote the development of a biofilm by growing B. subtilis in the biofilm-inducing medium, MSGG. After biofilm formation, the cells from the biofilm are dispersed and fixed with paraformaldehyde. The final step of the procedure is to detect the fluorescence signal emitted by single cells of the microbial community using fluorescence microscopy and flow cytometry. Ultimately, the results show how cells specialized to produce and secrete the extracellular matrix that constitute the biofilm are a different subpopulation from the cells that secrete surfactin, the signaling molecule responsible for the differentiation of the subpopulation of matrix producers.
The main advantage of this technique over existing methods like quantification of transcriptional expression using beta-galactosidase assays of microarray analysis is that it allows us to monitor gene expression at a single cell level and visualize particularly small subpopulations of cells that are specialized to express a specific gene. These gene expressions will not be detected when analyzing the overall population.
Integrative plasmids are used for labeling B. subtilis, and an example is shown here. PTAP-A, the promoter of the genes responsible for the production of TAS-A matrix protein, is cloned into the vector using the restriction sites EcoR1.